Hello, I'm Marco Molari. I'm a postdoc in the lab of Richard Neher in the University of Basel. And with this short video, I want to give you a brief introduction to PanGraph, a tool that we developed in the lab for the study of bacterial pangenome. PanGraph is able to build, given a set of microbial genomes, a compressed representation in the form of a graph. So what is the motivation behind this tool? The idea is that the microbial pangenome evolves under the frequent action of recombination and gene exchange. And if I want to know how a set of genomes are related, it's often not sufficient just to look at alignment of core genes, but it's important to look at the broader diversity. And this diversity can come in different flavors. Let's say that I have an isolate, I might have a gene that I'm interested in study. I might want to be able to stratify my population by presence-absence of this gene. I might also want to have the alignment of the region, and I also might want to know where this gene is present in the broader structural organization of the genome. And the pangenome graph is a representation that captures all of these aspects at the same time. So this tutorial is structured in these different parts. First, I will briefly explain what is a pangenome graph in our representation. And then I will give you a hands-on guide on how to use the command line interface to assemble a given set of sequences into a graph and exporting it in different formats. And I will show you a bit of the command line interface of PanGraph. This is by no means a complete exhaustive tutorial. There are more details in our documentation. I will put the link in the video description. And at the end, I will also give you an overview of some more examples that we added into this repository to give you some practical cases of use of PanGraph to say something interesting about a set of genomes. So let's start. So what is a pangenome graph in our representation? As I told you, this is a compressed concise representation for a set of genomes, that is, it's composed of two elements. One are blocks. So blocks are basically alignments. They have a consensus and they also encode variations for every particular instance of my sequence. So in a way, this representation is lossless. I can always reconstruct the particular sequence given the block. And the second elements are paths. So paths are instead a concise representation of a genome, not in the form of a sequence of nucleotides, but in the form of a sequence of blocks. So since I'm considering bacterial chromosomes, I imagine that these are circular, so you can imagine that genome A will start from here with block 1, then block 2 will follow, block 3, block 4, and block 5. And you also have to imagine that these blocks, of course, have a strandedness, have an orientation. A pangenome graph is nothing more than this collection of blocks and paths. I hope I can convince you that within this collection we have information on all of the layers that I talked about before: presence, absence, the alignments, and also the organization of the sequence. And we also believe that this structure encodes evolutionary events in a very natural way. You can imagine that if this small chunk of sequence got inserted recently, this is equivalent to a small loop in our graph where all of the sequences go through, all of the paths go through, except for one, which takes a small detour. Okay, so now let me show you how to build a graph. Okay, now for the more hands-on part of the tutorial, I prepared a folder with 5 FASTA files. These contain 5 chromosomes of E. coli sequence type 131 isolates that we want to compress into a graph. I will assume that you have PanGraph installed and available on your path. If not, we have instruction on our documentation on how to do this. So we will need the build command to construct our pangenome graph, and we can print the usage of the help command in this way: pangraph help build. So this will give us a description of all of the options. In particular, we will need to type pangraph build. We will need a circular option because we are dealing with circular chromosomes. Another important parameter is beta. As described in our paper, this parameter sets a threshold divergence for sequences that will be merged in the same block. In particular, its inverse represents the threshold divergence. So I will set this to 5 to have a threshold divergence of around 20%. And similarly, I will set the sensitivity of the aligner to 20. Then I will provide the data as input and pipe the results in a pangraph.json file. And PanGraph will start compressing these genomes into a graph. In the meantime, I can explain briefly how this works. So given our sequences, we will build a guide tree using alignment-free distance. This is done very quickly. And we will put our genomes as leaves of this tree. We then propagate the genomes along the tree, performing a merge at each intersection, a merge of sequences and a merge of graphs, and collecting the final pangenome graph on the root of the tree. And notice that all of the independent merges are performed in parallel by PanGraph. But what is the structure of the output file? The output file is a JSON file, as I was telling before, and it contains two main entries, which corresponds to blocks and paths. So blocks, as explained before, have a consensus and encode the alignment, and paths are our list of blocks. So in our documentation, we have more details on the content of these JSON files. In particular, blocks have an ID, which is randomly generated when they are created. They have a consensus, and they also encode the alignment in form of gaps, mutations, insertion, and deletions over this consensus. How this is done in details is again explained in the documentation, and even how we go from the consensus sequence to a particular entry in our alignment using these gaps, mutations, insertions, and deletions. And in the same way, paths have an ID corresponding to the ID of the FASTA file entry, and then have a list of blocks. In the meantime, PanGraph completed. This timer is not completely reliable, but it takes around 1 minute to build a graph for these 5 isolates. And this produced this pangraph.json file. This is around 6 megabytes in size. And if we compare this to the size of our FASTA files, we see that we are able to compress 5 isolates in something that has the size comparable to a single isolate, because these genomes are very similar and can be succinctly compressed into a graph. Okay, PanGraph also provides ways to export this graph into various formats for downstream analysis. One of these formats is GFA, which is a format that can be visualized with software such as Bandage. I will give you an example. This can be done using the export command. I will give the no duplication flag because we will avoid exporting duplicated blocks. And then I will provide my PanGraph file. This created an export folder, which a FASTA file and a GFA file. The FASTA file contains a list of entries, each one corresponding to the consensus sequence of a block. So you see here the ID of a block and its consensus sequence. And this for all of the blocks in my graph. The GFA file instead can be visualized with software such as Bandage. So let me quickly launch Bandage and load the graph. Okay, this is our graph. You see that since these genomes are structurally very, very similar, this is basically a circle with some variations around the edges when some private gene is present in, or private parts of the sequence are present in one of the isolates, but not the others. As a last example, I can show you the usage of the marginalize command. This command can be used to project a graph containing many, many strains on a subset of strains. which can be useful when the study of a particular subset is of interest and is much faster than building a graph from scratch for this strain. So in this case, it's simply sufficient to write PanGraph marginalized and then give a set of strains. In our case, I already pre-selected 2 isolates that are part of our graph, and then I give the graph as input and I pipe the results in another JSON file. And this created a marginalized graph, which is slightly smaller than the original one because it only contains 2 of the isolates. Okay, I just gave you a very hands-on tutorial on how to use PanGraph, but the question is, how can we use it to say something interesting about a given set of genomes? And to this end, we provide a more in-depth tutorial in which we analyze a data set of E. coli sequence type 131. This tutorial is divided in 3 parts, and I will briefly outline them. In the first part, what we do is we look for occurrences of gene beta-lactamase class CTX-M15 on the chromosome. And for every occurrence, we extract from the chromosome the gene along with a 5 kilobase pair upstream and downstream region. We then build a pangenome graph on these extracted regions, not on the full chromosome, but just on this region. And then we look at the structural diversity around our gene. So our gene is present here on the orange segment, and you can see that some of the structural diversity, the structural diversity allows us to define some relatedness between these occurrences of the genes. And in particular, we will relate the core genome divergence of the isolates with the structural diversity of the region surrounding the beta-lactamase gene. So here, for example, this blue block contains an insertion sequence that has duplicated itself many times on this region. In a second part of the tutorial, we will then look at, we will build a graph for a subset of 10 isolates, and then look at the amount of sequence that is private to one of the isolate in pairwise comparisons. The idea being that these genomes are very closely related if I look at the core genome alignment, but they have lots of sequence, several hundreds of genes that are present in one isolate but absent in the other, with a weak correlation between distance on core genome tree and private sequence, amount of private sequence between isolates. We will then zoom in a particular pair. So you see these isolates are close on the core genome tree, but they are quite different when it comes to amount of private sequence. And we will build a marginalized graph on these 2 isolates and compare the projection. So here you have a representation in which the common backbone is in the middle, and deviations from the common backbone represent part of the sequence that are private to one isolate or the other. And you see that one of these isolates contain big deletions or insertions, depending on which isolate we are considering. And colored segments represent duplicated blocks. So these are segments such as the insertion sequence we talked about before, and we have reasons to believe that around these segments more structural diversity should be present. So in fact, they often correspond to deviations of the common backbone. Okay, that's it for this tutorial. I will leave all of the relevant links in the video descriptions. Thank you for listening.